Welcome everyone to the RUNX1 Research Program hosted webinar series. Thank you all for joining us. I wanted to first spend a few moments just sharing with all of you, especially if any of you don't know us very well, a little bit about our organization. I'm Katrine Erickson. I'm the Executive Director of the RUNX1 Research Program. We are a not-for-profit organization, and we're dedicated to patients with RUNX1 familial platelet disorder with a predisposition to blood cancer. We do this, our work is really through raising awareness about this rare disorder. We are building a patient community that drives connection not only with each other, but also hopefully we can further integrate our patient community with our research community. And this is one way in which we want to implement that, and I'll speak about that in a moment. And then research funding. We fund quite a bit of research, not only independently as an organization, but also with our partners, Alex's Lemonade Stand Foundation and the Leukemia Lymphoma Society. And really what you should know, our mission, our research mission is really dedicated to finding and discovering interventions that prevent the onset of blood cancers in these patients. So I wanted to share with you before I introduce our speaker, and our speaker is actually today, I wanted to share with you that we will be hosting more of these webinars in the coming months, not only because we think it's a really efficient way to stay connected with the research community, but also because we're in a very new situation as a world with this pandemic. And we think it's important to maintain connection through this kind of collaboration and opportunities to share data. So please do check your email inboxes for future speaker announcements, and we hope to see you again. I wanted to also share with you that we have mostly scientists on the call today, but we do have a handful of patients. And as I mentioned, we're working really hard to build our patient community, and we want our patients to have a voice in research. And so patients have joined the call today. There's a handful of patients, and so we're going to structure the webinar as follows. So we'll start with a presentation by Dr. Speck and her student, Dana Bellissimo, and then we'll end with Q&A for scientists first, and then it'll be followed by a mini session for patients. So what I'd ask you to please do is make sure that you use the Q&A icon instead of the chat icon to ask your questions, because it helps us organize all the questions and allows Dr. Speck and Dr. Bellissimo to look at those Q&A questions and make sure that they answer each of them in order. So without further ado, I would like to introduce Dr. Nancy Speck, who is the chair of our Scientific Advisory Board, and as many of you know, a preeminent leader in the RUNX1 field, as well as, of course, the hematology world. She is been a great friend to our organization, and we're very grateful for all the time she's committed, not only to RUNX1 research, but also to our organization. So she's today going to share with you some of the work that actually RRP has funded. And this work is really important for many reasons, and I'm sure she'll help explain why it's so important. The work today is titled RUNX1 Mutations and Their Impact on Inflammation. This was recently published last week in Blood Advances, so please go ahead and check it out. And I'll turn it over to you, Nancy. Thank you.
Thanks, Katrine. It's been a real honor to work with your foundation, and it's been so impressive to see what the RRP has accomplished in such a short time. So welcome everybody, and thank you for participating in this webinar. I hope you're all well. The seminar today is going to be presented by Dr. Dana Bellissimo, who's a very talented MD PhD student who recently graduated from my lab. And as Katrine said, she published the work she's about to present last week in Blood Advances. So as a brief introduction, Dana obtained her BS in 2011 from the University of Wisconsin, where she majored in biochemistry, worked in Tom Record's lab on the thermodynamics of RNA polymerase. She spent a summer at the Wellcome Trust Sanger Institute and has a middle authored paper from that time on the genome-wide occupancy of 4 transcription factors, one of which was RUNX1 in megakaryocytes. And Dana joined my lab and Gary Gilliland's lab in 2014, and she graduated with her PhD in November of 2019. So one part of the story Dana won't share with you today is how she became interested in inflammation. So Dana's original project was to determine the mechanism by which mutations in RUNX1 and in another frequently co-mutated gene in AML and MDS, the gene called ASXL1, cooperated. So she made a mouse model in which she knocked out both genes in blood cells. And we were initially very encouraged that the mice with both genes knocked out were rapidly dying, but then became very perplexed when, after the mice were moved to a different room in our facility, the latency and penetrance of the disease decreased dramatically. And since the only difference between the mice was the room in which they were housed, we suspected the differences in the environment, and specifically in the microbiomes that the mice had acquired in the 2 rooms, were influencing the penetrance of the mutations. So Dana determined that the bone marrow from the mutant mice hyper-responded to bacterial ligands by overproducing inflammatory cytokines, and that the predominant contributor to the hyper-inflammatory response was the RUNX1 mutation and not the ASXL1 mutation. And so this launched us into what for us was an entirely new direction, which was to determine how RUNX1 controls inflammation. So with that, I'm going to turn this over to Dana for her to present to you this story. Dana?
Sorry about that. Doing a little negotiation with roommates over usage of the Wi-Fi for video conferencing right now. So I'll just bring up my slides now. And so I will dive right in. And so Nancy says I'm going to be presenting today my work that I did for my dissertation in the lab, and showing basically that RUNX1 had this really surprising role in regulating inflammation. And so as everyone on this call is probably well aware, RUNX1 is a key hematopoietic transcription factor that's mutated in sporadic and inherited forms of acute myeloid leukemia. And you can see it's frequently mutated in patients, shown here by data from the Cancer Genome Atlas, or the TCGA. And although there has been a lot of work done by our lab and many other labs, it was still unclear when I started my thesis work how exactly RUNX1 mutations function to promote leukemogenesis. And understanding how any mutation works in leukemia is really important, especially when you're thinking about trying to target these gene mutations in patients. And so here I'm showing a very simplified schematic of how leukemia occurs over time with the accumulation of mutations in the hematopoietic stem cell, or HSC, and then with successive accumulation of early initiating mutations that can contribute to a phenomenon known as clonal hematopoiesis, and then later accumulation of cooperating mutations. You can get frank progression to AML. And this clonal hematopoiesis phenomenon is one that was sort of recognized early in my PhD years, and it's where you have these mutations in leukemia-associated genes such as DNMT3A, TET2, and ASXL1. And you can see expansion of these clones in the blood of patients, especially those who are older, about 10% of people over the age of 70. However, they still contribute to quote unquote normal mature blood cell lineages, and only with the rare addition of multiple mutations in these HSC populations do you get frank transformation. And so really there have been a lot of sequencing studies to understand what these mutations are, and as well as to understand when they occur in patients. And some work has been done to target specific gene mutations, including FLT3 and IDH1. However, their clinical impact is somewhat limited, and they have to be combined with other agents. So really in understanding how mutations function in this process to allow us to target those is something that of great interest to the field. However, the the concept of targeting mutations in these patients becomes a little bit more daunting than when you look at the data and see that AML patients really have unique combinations of mutations. So here again, I'm showing data from the TCGA. And you can see here, this is every single column is a single patient. And the colored in bars are associated with gene mutations. And these gene categories over here on the left. And you can see here in red, I've highlighted those sections that RUNX1 mutations occur in. And so patients on average actually have a pretty small number of mutations, on average about 5 mutations per patient. And you can see here that they have really unique combinations of mutations, even though you can see some clear patterns of things that tend to co-occur or exclude. And so this individuality of patient mutations is really really a hurdle for the field, and pretty daunting when you're thinking about trying to specifically target any of these genes in patients. And so we wondered whether or not there was a common process instead that we could target in patients who have these unique sets of mutations. And so with this, sorry, I just got a notification my internet was unstable, just wanted to make sure things were going okay. So in turn, thinking of a common process that was mutated, we thought inflammation could be a really intriguing target, and it's really come to the forefront of hematopoiesis. And so there's a known role for inflammation in leukemia, and this has been evidenced by looking at alterations in serum cytokines of patients who have both clonal hematopoiesis as well as acute myeloid leukemia. There's also evidence of increased constitutive NF-kappa B activation in about 40% of AML patients. And this comes with potential functional consequences, as pro-inflammatory cytokines have been shown to promote the selective expansion of AML hematopoietic stem and progenitor cells, or HSPCs, to the detriment of normal HSPC growth. And the mechanism underlying this increased inflammatory signaling in AML is not what you would expect. It's not a result of mutations in the NF-kappa B pathway components themselves that have activating properties. And rather it's through other mechanisms such as altered expression of these inflammatory pathway components or expression of alternative isoforms of these pathway components. And so we wondered, given that inflammation is such a common feature in these patients, that perhaps more classic leukemia-associated genes may actually have a role in regulating inflammation in a way that just hadn't been previously appreciated. And so we wondered whether or not RUNX1 may actually play this role in regulating inflammation. And our first hints that this may indeed be the case actually came from those patients who have inherited forms of RUNX1 mutations. As I'm sure this audience is very well aware, this is familial platelet disorder with predisposition to acute myeloid leukemia. And in these patient pedigrees, there are signs that there's altered inflammation in these patients. So in some pedigrees, affected individuals also have eczema, and the severity of eczema correlates with the severity of the platelet defect. And further, those patients who have the worst eczema phenotype also tend to be the ones who develop leukemia. Further, it's been shown that in some of the first cohorts that have been enrolled in the NIH longitudinal study, there's a high prevalence of reactive airway disease and eczema. And so this made us wonder whether or not RUNX1 is actually playing a bona fide role in regulating inflammation. And so a further dive into the literature has shown that in cell lines, there's some conflicting results as to whether RUNX1 represses or augments NF-kappa B signaling. And the proposed mechanisms in the literature act through direct binding of RUNX1 to components of the NF-kappa B pathway members itself. There's also evidence that deletion of RUNX1 in lung epithelium of mice also results in constitutive NF-kappa B activation and increased susceptibility to LPS-induced acute lung injury. So there are some signs that RUNX1 may play a role in regulating inflammation, and so we really wanted to interrogate this question more fully. And so to do this, we used a conditional knockout mouse wherein RUNX1 alleles are floxed. And using a VAV1 CRE system, we can delete these alleles specifically in the hematopoietic compartment. And we focused in on myeloid cells and the innate immune signaling responses in the cell population, primarily because RUNX1 also plays a known role in regulating lymphopoiesis, especially at early lymphopoiesis stage. So these mice are relatively replete of lymphocytes. So first we looked at whether or not there were alterations in the makeup of the myeloid compartment of these mice. And so we can see here that looking at control, always shown in black, and the RUNX1s shown in green, that these mice have normal frequencies of monocytes and neutrophils in the bone marrow. To test the functional aspect of these myeloid cells, we utilized lipopolysaccharide, or LPS, which is a component of gram-negative bacterial cell walls, which can be found in both pathogenic bacterial species and also in those bacteria that make up the endogenous microbiome. And LPS is a really potent inducer of toll-like receptor 4 signaling. Toll-like receptor is a broad family of receptors that respond to specific molecular patterns. And activation of this receptor stimulates myeloid cells to produce pro-inflammatory cytokines, type 1 interferons, and chemokines. And the main producer of these high levels of secreted factors are the monocyte compartment in a wild type mouse. And so we wanted to know whether or not cytokine production was altered in our mice. And so we harvested whole bone marrow, stimulated with LPS for 8 hours, and then harvested supernate. And using BD's cytometric beta array, we were able to quantify cytokine levels in a multiplex fashion for 17 different factors. And what we saw is that several inflammatory cytokines were altered in the RUNX1 bone marrow, specifically upon induction of LPS stimulation. And so here I'm showing the cytokines that were altered. This is the absolute quantification of those levels in picograms per mil. And you can see here in this assay and many of the assays I'll show you here today, there's really no activity in the vehicle treated cells. So they're very quiet at baseline. It's only with addition of LPS that you see induction of this inflammatory secretion. And so you can see here for GCSF, IL-10, KC, also known as CXCL1, MCP1, and TNF-alpha, that you have really robust expression of these pro-inflammatory cytokines, specifically with induction with LPS and the RUNX1 knockout bone marrow. And we were specifically intrigued by TNF because of its known effects on in hematopoietic diseases and on HSC function, and also because simply it was the cytokine that we saw produced to the highest level in the RUNX1 knockout bone marrow. And so that previous assay was being done with bone marrow cells that are a heterogeneous mix of different cell types. And so we wanted to know which cells were responsible for producing these increased inflammatory cytokines. And so we harvested whole bone marrow once again, stimulated with LPS for 4 hours, and then harvested cells instead of supernatant, stained them extracellularly for our myeloid markers. This allows us to identify the different cell populations. Then we fixed and permeabilized the cells, and this allows us to stain intracellularly for cytokines. And in this assay, we utilized a TNF-alpha antibody for which we can get very good staining. You can see here in the monocyte compartment, you get really nice bright staining for TNF-alpha. These cells produce a lot of TNF-alpha in this assay. However, we saw no significant differences in TNF-alpha production by the monocytes. However, when we looked to the neutrophils in this assay, that's where we started to see significant and interesting changes. So you can see here that a normal neutrophil that when stimulated with LPS produces, there are a low frequency of neutrophils that produce low levels of TNF-alpha. However, RUNX1 knockout neutrophils actually produce robust levels of TNF-alpha. And you can see this increase both by looking at the percentage of TNF-alpha positive neutrophils, which is markedly increased in the RUNX1 knockout mice. And also, we can see that these neutrophils are actually producing more TNF-alpha on a per cell basis based on this increase in median fluorescence intensity, which is a measure of how bright the staining is. So there are more neutrophils producing TNF-alpha at a higher level, making neutrophils a really strong candidate for the source of these increased inflammatory signaling molecules that we're seeing in the RUNX1 knockout bone marrow. And so the intracellular flow antibodies are somewhat limiting for how many cytokines we can analyze. And so we harvested bone marrow from our mice and sorted neutrophils, and then again utilized this BD stadiumetric bead array to look at a full complement of 17 different factors simultaneously to see what was altered in the isolated neutrophils as well as isolated monocytes. And in this assay, we plated the same exact number of cells into every well in the same volume so they can kind of compare on a per cell basis the levels of cytokines that are being produced. And again, you see that when in this assay with isolated neutrophils, when you stimulate with LPS, again, the RUNX1 knockout neutrophils make high levels of TNF alpha. There's also a slight but statistically significant increase in IL-1 alpha, as well as a robust increased secretion in the chemokine MIP-1 alpha. And in thinking about the levels of this TNF alpha that's being produced by neutrophils, it's particularly striking when you compare it to the monocytes sorted from the same mice. So you can see that monocytes also produce high levels of TNF alpha upon stimulation. But when you compare these levels, the RUNX1 knockout neutrophils are making levels of TNF that are similar to or even higher than normal monocytes. And when you think about the relative frequencies of these cells in the bone marrow, this can be, we think this is really important. So the neutrophils make up about 50% of a murine bone marrow, whereas the monocytes make up less than 10%. So now you have this new source of high levels of TNF alpha in a cell population that's ubiquitous in the bone marrow. So we really think this is a very biologically significant contributor of pro-inflammatory cytokine responses in the bone marrow. And so next we had been specifically looking at toll-like receptor 4 stimulation, and I remember I mentioned that toll-like receptor 4 is just one of a family member of toll-like receptors. So next we wanted to see whether or not this was specifically dysregulated or whether toll-like receptor signaling was dysregulated more broadly. And to do that, we assayed all of the toll-like receptor agonists shown here in blue and used our same intracellular flow assay to look at neutrophil responses. And so we can see here that when we harvested bone marrow from our mice and stimulated and did the same intracellular flow assay, that in fact the responses are quite specific. So these neutrophils produce increased levels of TNF alpha, specifically with LPS stimulation and PAM3CSK4 stimulation, which is a toll-like receptor 1/2 agonist. So we see this dysregulation leading to increased downstream inflammatory cytokine production specific to toll-like receptor 1/2 and toll-like receptor 4 stimulation. And so given that we were seeing these inflammatory consequences downstream of a toll-like receptor agonist, we wanted to look at the signaling pathways that mediate this inflammatory response. And so we looked at the TRAP MIT88 arm downstream that leads to activation of NF-kappa B. And so normally NF-kappa B is held inactive in the cytoplasm by the inhibitor of NF-kappa B or I-kappa B. However, upon activation of these upstream signaling arms, you get phosphorylation of I-kappa B. This leads to proteasomal degradation, releasing NF-kappa B to translocate into the nucleus and activate an inflammatory cytokine response, including transcription of its own inhibitor I-kappa B as part of an auto-regulatory feedback loop. And so experimentally, we can use I-kappa B degradation as an experimental readout of activation of this arm of the toll-like receptor signaling pathway. And so what we saw both in whole bone marrow and in isolated neutrophils, which I'm showing you here, that when you stimulate with LPS, you can see some degradation of I-kappa B over time. And this degradation is more rapid and more marked in the RUNX1 knockout neutrophils, which is quantified here. So we can see that there indeed is increased activation through the TRAP MIT88 arm of the toll-like receptor signaling pathway in these neutrophils. So all the assays I had done up to this point were really looking at ex vivo functions of neutrophils from bone marrow, and we really wanted to know whether or not there were biologically significant effects in vivo. And to do this, we utilized an LPS inhalation model. So in this model, you use a single chamber that houses the control in RUNX1 knockout mice, and they breathe in nebulized LPS for 30 minutes. And then 24 hours later, you harvest the lungs for both histology or for bronchoalveolar lavage or BAL, and this is where you flush a fluid into the lungs and to see what cells and cytokines were present in the lung microenvironment. And you can see here by looking at the cytospins of the BAL fluid that this is a great assay for looking at neutrophil activation, as the majority of these cells that you're seeing here are these polymorphonuclear neutrophils or PMNs. And there's just a smattering of these fried egg appearing alveolar macrophages. And you can see here by eye, which is quantitated above, there's actually an increased infiltration of neutrophils into the lungs of the RUNX1 knockout mice. So you get a significant migration of neutrophils into the lungs of the control mice after this LPS inhalation. However, this is almost 50% higher when you quantify the numbers of neutrophils in the BAL fluid. So you really do have this increased inflammatory infiltrate. And this increased inflammatory cell population is not without consequences, as we can see here from just the appearance of the BAL fluid, which is frankly bloody, specifically in the RUNX1 knockout mice, is the sign of really profuse alveolar hemorrhage. You can also see this here by looking at histology from mice performed on the same day. And you can see here, although there is signs of inflammatory infiltrates and some damage to the lungs in the control mice, there's just frank alveolar hemorrhage and inflammatory damage in the lungs of these mice. And it's also important to note that likely contributing to this phenotype may be platelet defects, because we're using a pan-hematopoietic deletion of RUNX1. And this damage is occurring in an environment full of inflammatory cytokines. So you can see here that when we quantify the cytokine levels in the BAL fluid that we get from the lungs of these mice, there are many different pro-inflammatory markers are markedly increased in these mice, including GCSF, the IL-1 family members, chemokines, as well as reassuredly, TNF levels are also increased in the BAL of these mice. So we think clearly that these increased inflammatory responses to the neutrophils have significant biological effects in vivo. So the next question we had is, why is toll-like receptor signaling dysregulated in the RUNX1 knockout neutrophils? And given that RUNX1 classic role is as a transcription factor, we wanted to see whether or not the transcriptional state of the RUNX1 knockout neutrophils was dysregulated. And so we harvested bone marrow, once again, sorted neutrophils, stimulated for 2 hours with LPS, and then harvested these cells for RNA-seq, utilizing a spike in control to normalize for cell number. And what we could see immediately in the gene ontology analysis, or the GO analysis that we did, was inflammatory pathway activation, as you can see here in many of these terms. And this was true even in the vehicle-treated neutrophils. So these are neutrophils at baseline that haven't been stimulated. And we're seeing terms such as toll-like receptor signaling pathway and positive regulation of NF-kappa B signaling as upregulated at baseline in the RUNX1 knockout neutrophils. And so when we curate all of the toll-like receptor pathway components as a gene list and look at whether or not their expression is significantly upregulated, we do see that that is in fact the case over the 3 replicates of these RUNX1 knockout neutrophils that were vehicle treated, so their expression at baseline, we see a significant increase in expression of the toll-like receptor pathway overall. And here I've annotated in green all of the genes that were significantly upregulated in the RUNX1 knockout neutrophils. And you can see here that it really is a broad range of toll-like receptor pathway components, including toll-like receptor 1 and cells themselves, adapters, upstream signaling pathways, as well as NF-kappa B family members themselves, P50 and P65. And we also were able to validate that toll-like receptor 4 protein levels are indeed upregulated on the surface of RUNX1 knockout neutrophils. So this data really shows that it seems like there is a transcriptional program that upregulates many of the components of the toll-like receptor pathway, and this is likely what's leaving these cells primed to hyperrespond to stimulation of these pathways. So next we wanted to know whether or not RUNX1 is directly regulating toll-like receptor signaling in neutrophils. Again, we've been using a pan-hematopoietic knockout of RUNX1, which could have influences of other cell types. And so we really wanted to isolate RUNX1's function to the neutrophil compartment. And so we utilized an MRP8-CRE model, which deletes RUNX1 specifically in the neutrophil compartment. Again, these are also known as polymorphonuclear neutrophils, or PMNs. And so I'm going to refer to these as the RUNX1 PMN knockout mice. And so we did our same assay with RUNX1 specifically deleted in the neutrophil compartment, and we're surprised to see that in fact that our inflammatory phenotype disappears. So if you look at the percentage of neutrophils that are TNF alpha positive after stimulation with LPS, you no longer see this dramatic increase, but in fact these RUNX1 PMN knockout neutrophils that are shown here in purple look like control neutrophils. Again, they're producing a low frequency of neutrophils is producing low levels of TNF alpha. Further, when we look at the brightness or the indication of the level of TNF alpha standing by the median fluorescence intensity, once again, these cells look like controls. So when you lose RUNX1 in the neutrophil compartment, you have no effect. And perhaps we shouldn't have been so surprised that this was in fact the case, because RUNX1 expression is markedly downregulated during normal differentiation of neutrophils. And so here you can see here in this protein plot that even in the control neutrophils, we're unable to detect RUNX1 protein, although we could readily detect it in the spleens of the same mice. And this really matches up well with transcriptional data that's been published by other groups that shows that RUNX1 is really expressed highly in early progenitors, such as the granulocyte macrophage progenitor, and then is markedly downregulated throughout neutrophil progenitors and neutrophil commitment. So perhaps it's not so surprising that when you lose RUNX1 function in these later neutrophil stages, that you're not getting an effect. And this led us to hypothesize that either RUNX1's effects are being established earlier in this myeloid progenitor population upstream of neutrophil commitment, or from an alternative cell type. And so we really wanted to pin down whether or not RUNX1 was having its effect in an upstream myeloid progenitor, and to determine when exactly in myeloid development was this inflammatory transcriptional program established. And unfortunately for us, the RUNX1 knockout mice really like to misexpress all of those key markers that you would use to identify the different hematopoietic stem and progenitor cell populations, including the ones you would use to purify GMPs. So we really utilized single cell sequencing technology to identify these cell types based on whole gene expression programs instead of a few select markers. And so to do this, we harvested bone marrow from our mice and looked at lineage negative c-KIT positive SCA negative cells shown here. And these contain your hematopoietic progenitors such as the GMP, CMP, and MEP, and then performed single cell sequencing analysis. And through the help of our collaborators, we were able to map our populations onto the data from Gelati et al. From Ito Amit's group, wherein they used index fax sorting, as well as single cell RNA-seq in order to correlate sort of those canonical markers we use to identify these populations by flow cytometry with our gene expression patterns. And this allowed us to identify the different cell types in our populations from this study using gene expression rather than those markers that we were uncertain which populations they were identifying in our mice. And you can see here that we get good coverage of the spectrum from the control and the RUNX1 knockout mice, and this is only sorting a subset of the cells that were fully characterized in this paper. And we're really just going to focus on this myeloid arm of this pathway from the orange CD34 early hematopoietic stem and progenitor cells through these GMP, like in pale blue, CD34, CD16 populations, and these further neutrophil progenitors in lavender and fuchsia down to the neutrophil. And so what we saw that was when we looked at differential expression comparing the control and RUNX1 knockout mice, shown here, looking at those terms that were upregulated. Again, we see lots of different pro-inflammatory signaling pathways that are upregulated in these early hematopoietic progenitors. And if we look at the expression pattern throughout differentiation, so again, from these early hematopoietic stem and progenitors down through these more mature neutrophil progenitors, you can see these and then cluster those pathways based on their patterns of expression, you can see that in these terms that become more upregulated as these cells differentiated, the regulation of toll-like receptor by endogenous ligand and toll-like receptor 4 cascade pop up in this analysis. And if we look at this toll-like receptor 4 cascade and assign it an activity store, we can really see how this, this program turns on from the CD34 early hematopoietic stem and progenitor stage to this GMP-like stage. It's become significantly upregulated in the RUNX1 knockout cells at this GMP-like stage, and then is maintained throughout differentiation, presumably to those neutrophils that we analyzed in the bulk RNA-seq. So we can really see from this data that this program is turned on early in myeloid differentiation and then maintained. And so we think that RUNX1 is actually acting to establish this neutrophil phenotype early. So in the model that we would propose, RUNX1's function is actually indispensable for its direct regulation of neutrophil activity in these later neutrophils, but instead is acting in an early hematopoietic progenitor, likely a GMP-like stage, to activate this increased transcription of these toll-like receptor pathway components. This transcriptional program is then maintained in differentiated neutrophils that leaves them primed to hyper-respond to pro-inflammatory signaling that creates this inflammatory cytokine response that has biologically significant consequences, including tissue damage. And so we think this is really important when you think about patients, especially patients with FPD-AML, in which all of their cells have RUNX1 mutations, shown here by these green stars. And in these patients, their hematopoietic stem and progenitor cells have RUNX1 mutations, and their differentiating myeloid cells, including their neutrophils and macrophages, also have these mutations. And we would suspect, based on our data, that perhaps their differentiating neutrophils also have these increased inflammatory responses. And maybe this establishes an inflammatory niche that can select for the expansion of pre-leukemic or leukemic clones upon acquisition of secondary mutations. And so we actually think that this inflammation from differentiated myeloid cells due to RUNX1 mutations may actually be a key factor underlying the way that RUNX1 mutations actually actively promote leukemiogenesis in patients. And so this brings us back to the question that I posed at the beginning, is whether there is a common process that we can target in patients who have unique sets of mutations. So can we broaden out this view to mutations beyond RUNX1? And the example that we're most excited about is TET2, which is one of these early mutations that is seen in the process of clonal hematopoiesis. And as many people listening in may know, these patients who have clonal hematopoiesis their differentiated myeloid cells aren't quite normal. And in fact, these patients have increased risk of cardiovascular disease, and they've been shown to have elevated serum levels of IL-8, which is a canonical neutrophil chemokine that is expressed in humans but not in mice. In multiple groups in 2017 simultaneously published that they were able to recapitulate this aortic atherosclerosis, the pathogenesis that underlies cardiovascular disease in humans, using bone marrow that had TET2 either a single copy or both copies knocked out. So here, these are aortas from those mice, and they're stained with oil red O staining. And so you can see here these really enormous lipid-laden plaques in the aortas of even the heterozygous knockout mice. And they showed that this was associated with elevated secretion of inflammatory cytokines from myeloid cells. In this model, it was increased secretion from macrophages. And in fact, myeloid-specific deletion of TET2 was sufficient to accelerate our aortic atherosclerosis in these models. And so we think this actually looks an awful lot like what RUNX1 mutations do. So again, you have a mutation in a hematopoietic stem and progenitor cell that's leading to differentiated myeloid cells producing increased levels of pro-inflammatory cytokines in a state that predisposes to an increased risk of leukemic transformation. And so we think that, in fact, inflammation may be a common consequence of mutations in leukemia-associated genes, and perhaps it may actually be one that is targetable. And so with that, I've already gotten multiple shout outs about my paper that was published last week in Blood Advances. So happy to answer questions, but also I would refer you to that paper if you want to look more into the details. And actually thanks to Katrine for tweeting out about that, because that's actually how I first found out that it had made its way all the way to publication online. And so with that, I'd like to thank everyone who helped with this work, including my mentors in my very large and complicated lab families from the Speck, and Jordan labs, and specifically Lee and Samita, who in the Speck lab, who contributed directly to the work I showed here today. I'd also like to thank our many wonderful collaborators at Children's Hospital of Philadelphia, or CHOP, including hematopathologist Gerald Wertheim, who was the one who helped us make the transition from the phenotypic change that Nancy described that got us into this work, into looking at inflammation, as well as Scott Worthen and Ning in his lab, who helped with the really beautiful LPS inhalation assays into doing that in vivo model. And also our very close collaborators in the Speck lab, which is Kai Tan's lab, including Chin and Shiwei, who helped with the really heroic and speedy single cell RNA-seq efforts for the reviews for this paper, and Bing, who helped with the bulk RNA-seq data of the neutrophils. Also like to, of course, thank Penn and the core facilities there, the Penn MSDP program, and my funding, including, of course, the RUNX1 research program. And so with that, I think we're going to open it up to questions.
Yeah, Dana, can you see the Q&A? Do you want to run through, or Nancy, whichever one of you? There's quite a few questions.
Let's see.
I'm muted.
I guess I can just start from the top. So it looks like we have a question from Marty Ponce asking if we looked at mRNA levels in neutrophils, monocytes, and MEGs. And so we looked at the mRNA and the neutrophils, but we didn't do RNA sequencing or any sort of expression level in the monocytes or the MEGs. We really focused in on the neutrophils in this project, but we have hints through some of the work that I've done that myeloid compartments are also altered in these mice, and so I think there's certainly more work to do in the other myeloid compartments. Let's see. And from Diane Krause, we have a question. So only a subset of neutrophils in the bone marrow showed increased TNF-alpha in response to LPS. Do you have a hypothesis as to what makes this subpopulation of neutrophils unique? And is that the same with increased MIP-1-alpha? And so I think it's a little difficult to know exactly based on intracellular flow whether you can kind of get relative levels from that, but I can just tell by looking at other cytokines and chemokines that it's not necessarily the most sensitive assay for knowing exactly how many cells. So I wouldn't be surprised if there are subsets of cells that are more or less inflammatory. That has certainly been shown to be, there are different sort of more activated neutrophil subsets. Also in the gating scheme that we use, it also captures these sort of pre-neutrophils and early neutrophil progenitors, which have only sort of recently been described, so that could be contributing. And we haven't looked at MIP-1-alpha in these cells, so I don't know. I'm not sure if that would also be the case, but I wouldn't be surprised if there are slight differences in subsets and what they're producing. Okay, and then from Lin Han, we have, in total, you have more than 3,000 picogram per mil increase in TNF as shown in the serum assay, and you show neutrophils contribute to 1,000 picogram per gram increase. Do you expect other resources for this increase in TNF? And so, yes, there are other cell types that are making it. And also, these two assays, everything is having the same number of cells, so it's kind of on a per cell basis, but I think certainly some of this phenotype is from isolated neutrophils, but I think having those multiple cell types in the bone marrow, you're going to have feed-forward mechanism, you're going to have more pro-inflammatory cytokines and signalings. So I think it's not surprising that we see more effects in total bone marrow and that other cell types are also contributing. Let's see. There's a question about, another question from Marty Pons asking if we've tried the Flox RUNX1 with a platelet Cree and do the LPS inhalation. We haven't. We've been curious to tease apart what the effects are of neutrophils versus platelets, but we haven't gotten around to that yet. From Amy Fan, we have in ex vivo experiments. Have you removed LPS and see if they continue to be hyperinflammatory, such as a washout experiment? We have not. I think that's a really interesting proposal. I think it's also interesting to see, one of the experiments we toyed around was whether or not we do like low doses of LPS over time, whether there's a priming effect. So we've only really done the single stim and then harvest at different times based on what output we're looking for. Let's see. So I'm actually seeing several questions asking if we think there is an increased risk for patients with FPDML with COVID-19, and I'm not going to make any sort of claims about that. I think we've done like a single experiment with a patient sample that shows like maybe there are altered inflammatory responses. I think patients in general have really divergent inflammatory responses, especially when you look at their neutrophils. Just normal neutrophils have, from different humans have a huge range of how they respond to LPS stimulation and how much activation you get and how much inflammatory cytokine. It wouldn't surprise me if that's the case, but I'm not sure which cell types might be altered. I'm also not as familiar with the literature that's coming out and what cell types are critical for this. So I will leave the speculating at that.
Dana, you might want to talk about the question just before that from Shira Zwirin.
Okay, this is my daughter with a RUNX1 mutation was also diagnosed with suspected systemic juvenile arthritis. And
Dana seems to be frozen.
Can be helpful in patients with RUNX1 mutation to prevent the development of leukemia. My internet flagged as being unstable, but I just posted that I'm going to answer it live, so other people should be able to see the question in the Q&A as I answer it. And so this is actually something that I thought a lot about when I was presenting my thesis work. And we do think that if we can show that patients have alterations in these cytokines, that perhaps this is a way that we can target the leukemic progression in these patients. There's a lot more work that needs to be done, but we think that's a really interesting idea. In fact, there is evidence that in patients who have heart disease, they were given an IL-1 blocker to try and reduce their coronary artery disease risk, and they happened to find that those patients also had reduced levels of lung cancer. And so I think in patient populations where they are having being treated with a cytokine receptor blockade, there are some signs that you could see some effects on leukemogenesis. I think we still have a lot of work to show that mechanism. We think it's a really interesting idea and something that should be teased apart in the future. Let's see.
Why did you start looking at TLR4 in particular?
So the reason we started looking at TLR4 is just because we wanted to look at innate immune signaling in the myeloid compartment, which is really those first responder cells to inflammatory responses. And so they recognize these common patterns that you have on pathogens known as PAMSR. And so this is kind of one of the common ways that this compartment responds, and LPS is a very well utilized and potent agonist that's studied. And so we found a really potent phenotype with that and sort of went down that avenue, but that's also why we tested all the rest of the toll-like receptors as well.
Have you checked cytokines in stem and progenitor cells?
So I have tried to do the same assays. This is all limited by the same pesky problem that the RUNX1 knockout stem and progenitor cells have altered expression of those markers that you need to identify those populations. And so in order to sort of do those assays, we kind of have to go back and revalidate what those populations are in the RUNX1 knockout bone marrow, which we think the single cell sequencing will help with. But that's kind of why we went to that really brute force effort to look at total gene expression to identify the populations. I've done some attempts at doing the stimulation, but again, it's a little hard to interpret because we're not sure exactly what the populations are because the markers are expressed. But it looks like comparing some populations with the markers that we can use, there is some increased TNF alpha production, but certainly not to the level seen by differentiated myeloid cells.
Have you, or do you have any plans to determine the contribution of megakaryocytes in this process? This is from Bert van der Rijden. As these are overrepresented in RUNX1 patients and likely will leak cytokines?
I think that's a really interesting proposition. And we've talked with some of the other people who study RUNX1 and megakaryocytes. It's certainly not our lab's expertise, but it's something we're very curious about. And we think, especially with the lung inhalation phenotype and sort of known studies showing that sort of platelets and neutrophils and inflammatory responses can modulate each other's activities, we think that could have, that could be contributing here. And that's like another whole field of work. But we think this is, the neutrophils are really a producer of really high levels of these inflammatory molecules, but we don't think that they're the only population that's dysregulated and that might have altered inflammatory responses.
Paula Heller wants to know if we've seen increased plasma levels of these cytokines in the mice.
So in the mice, I think I did one experiment, and you don't see increased plasma levels at baseline, and really that's the case for everything that I do. The baseline expression of these the inflammatory cytokines is really quiet. So they don't seem to have much basal activity. It's only when you stimulate. And then in our stimulation assays, I looked at BAL, but I haven't looked at plasma or done any other sort of in vivo injections of LPS and looking at the plasma.
John Bushwiller would like to know if we have any data to indicate that the point mutations that arise in FPD AML give similar effects.
So we basically our hints are from the fact that we see these anecdotal and now gathering like more numeric evidence that patients have altered inflammatory responses by some of these autoimmune effects. We've done one experiment with one patient, their peripheral blood neutrophils, and we can see that maybe there are things that are altered, but also something that we saw because we had more control volunteers in the experiment is that, as has been published, there's a huge range in how neutrophils respond. And so I think we just need more patient data and lots of controls and to be able to make that answer. So there are some signs that perhaps, but I think based on a single patient and the amount of variability that exists in the population, I can't make any strong claims.
AAFAQ, did you see the level of platelets in the studied mice? Is there any role of platelets there?
So the platelet levels are a little bit decreased, I believe, in the RUNX1 knockout mice. And I do believe that there's likely a component of a platelet defect given the really frank hemorrhage that we saw. So when we were working with the collaborator, Scott Worthen, who does this assay all the time in different models of activation of inflammation, he has never seen anything like that. He thought that he may have gotten into like an artery, but then it happened specifically in only the RUNX1 knockout mice. And so I think that there are other factors playing a role, not just inflammation, but also if you have these platelet defects, you could see how those two things together would really lead to that hemorrhagic phenotype.
Yeah, I also would like to add to that lung study, this other group also knocked out RUNX1 in lung epithelial cells, as Dana mentioned in the beginning of her talk, and it got the same phenotype. So I think low levels of RUNX1 in multiple tissues are going to be contributing to these phenotypes. Lucio Castillo would like to know, you postulate inflammation selects for pre-leukemic clones. Have you evaluated if LPS promotes proliferation or survival of RUNX1 mutant clones or a negative effect on wild-type progenitors?
I believe LPS has over time a negative effect on progenitors, but I'm losing that piece of data off the top of my head. But we haven't looked specifically in the RUNX1 mutant clones. Again, I think part of the work is we had talked about doing those experiments, but we weren't sure when this phenotype was being activated at the time. The single cell RNA-seq data is really hot off the presses for us, and we also weren't sure how to isolate those populations in order to test it. It would have been very nice to have like sorted GMPs and put them in different cultures and see how they grew or differentiated. But because we didn't know what actually was the GMP in these mice based on published markers, it limited our effects. But it's certainly something that we've talked about and really would like to do is see if that occurs. Certainly in work from Anna Priya's lab shows that a good majority of this primary AML samples that she tested were sensitive to factors such as IL-1 beta. And so they had increased expansion specifically of the AML clones to the detriment of normal hematopoietic stem and progenitor cells. So I think there are signs from other parts of the literature that that might be the case, but we certainly need to test that in our model.
Alan Cantor would like to know whether patients with germline RUNX1 mutations have increased plasma inflammatory cytokine levels. Yeah, Katrine, do you know? Paul? Liu?
I believe Anu Priya is looking at that, but I don't think just in general plasma levels. I think it's more under stimulation that they see that effect. But I don't want to answer because I'm not quite sure what data she's collected on plasma levels.
Yeah, based on our complete knockouts, I would expect that it's with stimulation that you're seeing those effects. But we haven't looked at that in a germline knockout mouse model.
Fabiola.
Sorry to interrupt, Nancy. I just want to let the patients who are on the line know that we still want to have a mini discussion with you. We have a few more questions that we want to try to get through with the scientists, so I do hope you can stay on with us. Just hang in there for a little bit. Thank you.
Just 3 more questions. Fabiola de Carpiacano, what was the age of the mice where you obtained the bone marrow?
So these are all experiments done in mice 6 to 12 weeks old, so young adults.
And Samarakoti, do you think inhibitors of the TLRs, more specifically those for TLR4, would be able to rescue the overactivation?
I think that's a really interesting question. We're actually trying to get at that in the lab. An MD PhD student who joined the lab shortly before I left the lab is actually breeding mice for toll-like receptor 4 knockout. So we were going to see if you kind of reduce by half at first, if you can block from the receptor level, whether or not that it inhibits that effect. And then she's also looking into possibly doing some inhibitor experiments. I would expect that it would have some effect. I don't know if it would fully rescue, just because there's so many components of the pathway are upregulated. I don't know if you get like any stimulation through it, if you're still going to have some effect.
From Morty Pons again, another reason why the bowel was hemorrhagic was that the platelet alpha granules are full of cytokines and attract neutrophils to the injured lungs. Yeah. Did you measure cytokine levels in MEGs?
No, we unfortunately have not done any experiments in MEGs, though we think that is a contributor. But we're very neutrophil biased in our assays, so we certainly have thought about them, and it's an interesting experiment. I think, especially in the lung inhalation assay, the MEGs and platelets are playing a role that we're just not measuring.
And Morty, you can help us since you are right next door. That's all the questions that we have here. So thank you everybody very much for participating and for all the really great questions for Dana.
Yes, I just want to echo Nancy's comments. Thank you everyone for attending, and thanks for the rich set of questions, and we hope you join us again for our next webinar, so please stay tuned. I will send out emails announcing the next speaker very soon. And with that, I think those of you who are scientists are released. If you do not have to stay for the rest of this session, we're going to speak with any patients who have questions. So please, if you have any questions, patients, please type them in the Q&A icon. I think that's the best way forward given bandwidth issues rather than having a conversation, unfortunately.
The scientists, any of you who actually treat patients with FPD AML, if you would like to stay on the line to help answer some of these more specific questions, that would be helpful. Thank you.
That's a great idea. So please don't be shy. Don't see any questions yet. I know that there were some of you on, and maybe you did have to leave, so we'll give it a few more minutes, and then we can certainly, if those of you who did join us are interested, maybe we could even have a short session, set up another call in the future that's more private, if that's of interest. And maybe Nancy, you might want to just give a short summary of what you think this means for patients and future research and future directions.
So I'm very hopeful that reducing inflammatory cytokine could be a therapeutic option. I mean, as Dana said, we have a really long way to go to figure out whether that would be the case. I mean, we need to figure out what inflammatory how the hematopoietic stem and progenitor cells are responding to these inflammatory cytokines. Have we captured all of them? Are there other cell populations that are contributing? Which inflammatory cytokines are important and how do they affect the hematopoietic stem and progenitor cells? And then we can think about models whereby we could try inhibiting those cytokines and preventing disease progression. I mean, one of the problems in the field right now is we don't really have a good progression model for the leukemia. So that's hampering our efforts a bit. But there are multiple funded investigators who are trying to develop such a model.
Nancy, what do you mean by progression model, just so people can understand?
A progression model would be a model whereby you could introduce one mutation and so start with a RUNX1 mutation, maybe introduce another mutation or two mutations, and then over time, hopefully the cells would continue to accumulate mutations and develop into a frank leukemia. So a model that would capture the very earliest events all the way to the later events. And so if we have models whereby we can show that additional mutations cause an expansion of a hematopoietic stem cell clone, could we, by treating with inflammatory inhibitors, could we prevent that expansion, or could we reverse that expansion and eliminate that clone?
Thank you. So it looks like we actually have a question from Shira. And she says, I had already asked about my daughter with RUNX1 and systemic JIA, but I'm also wondering if it's possible for a RUNX1 mutation to cause the kind of inflammatory response that could mimic an autoinflammatory syndrome.
So I think it's possible. There certainly have been sort of GWAS or gene association studies showing that in patients that have a slightly different DNA sequences in genes, certain genes has been associated with autoimmune diseases. So you have GWAS hits with RUNX1 linked to those inflammatory processes. Certainly neutrophil inflammation is not the type of neutrophil inflammation that's specifically associated with sort of eczema or these other phenotypes. I think it's possible. I also just think we're kind of a little tunnel visioned on the neutrophils right now, and I think it would be interesting to look at other types of inflammation using more of an autoimmune type model, which is again something you kind of have to stimulate in the mice.
Thank you, Dana. There was another question asking, did I understand well, is there an association of RUNX1 mutation to eczema? Is that related to any age? Is there an age-dependent effect here?
I am not aware of an age.
So I think the data that we have.
Go ahead, Dana.
Eczema usually presents in childhood, and I think in the pedigrees from that single case report, there were pediatric patients in there that had the expression. So I would guess that it's eczema phenotype that starts early, and eczema is really common. So I think that's why this NIH longitudinal study is so important for us to really look at what other things are associated with this gene mutation, because for things that are common, it might take a good amount of data to see that there's actually a significant increase.
But my understanding from that study is so far there does seem to be a lot of reports of eczema, correct, yeah. Katrine?
Yes, yes. And we've learned from Paul, and who I believe was just on, but I'm going to allow him to speak because he is the principal investigator of the, excuse me while I try to figure out how to allow him to speak. He's the principal investigator of the natural history study, and so I think it would be great to hear directly from him.
Yes, it would be.
Paul, I just unmuted your line. Are you able to speak?
Can you hear me?
Yes.
Okay, great. Yeah, I wasn't able to unmute myself, and yes, you're right. And in our patient population, ectopic, reactions, eczema, asthma, and other, immune, not autoimmune, but allergy to food and other, stimuli are fairly common in our patient population, not only in children, but also in adults as well. And we also have seen patients IgE level increase, and increased number of eosinophils. If you're adding them all together, I think it's more than half of our patients. This is something we are really interested in, and we would like to, we are exploring further with immunologists on campus, and of course, with other, collaborators extramurally as well.
Great, thank you for that, Paul. Well, with that, I don't see any other questions from patients. So I'd like to close this webinar series and thank again Dana and Nancy for spending the time with all of us and sharing this really important work. And we look forward to future work coming out of the Speck lab and maybe a future Bellissimo lab. So thank you. And thank you everyone who joined. And as I said, please look out for the next webinar, and take care, be well, stay safe.
Bye everyone.
Bye.